Yes, they are.
Hello everyone, my name is Rohit and I am research scholar from Punjab University, Chandigarh from India, and I am working on murine rodents. So here I am going to present their phylogenetic affinities and biogeographical implications. So in this presentation, I will cover introduction part, previous work, objectives of my research and their significance, geology of the area, and ecology and results. So murine rodents are most species mammalian order. It comes under Muridae family, and this subfamily contain around 650 species. So in my study, I have included 350 species for my study and also collected some fossils from Indian subcontinent. So murine rodents are mostly old world rats and mice. These are divided into 15 tribes having 656 species and 350 135 genera, and most comes under subfamily Murinae. So, based on the fossil record, this family originated in Indomalayan region in most of the fossils found in Pakistan region in Shivaliks, which is Miocene in age. And this subfamily distributed all over Africa, Eurasia, extending to Australia and New Guinea and many islands. So previously, in 1969, Missoni distinguished Murinae family from Chrysitinae based on their dental pattern. But in 2009, Wessels was against such classification because this classification was based only on morphological characters. But after molecular data, this classification was failed because Acomys have dental morphological dental pattern which was similar to Murinae subfamily, but molecular data was not supported that classification. So oldest fossil of this family is in Pakistan region, which is 13.8 million year old, Antimus tingiensis, which is based on magnetostratigraphy. And previously, many people have worked on this family, Muridae, and many people gave many ages, which are around 12 to 23 million year old. So in this study, I have used tip dating method, and most of them have used node dating method. So based on this, from these fossils records, were expecting that this subfamily originated in Indomalayan region, which is Southeast Asia. So objective of my thesis is to assess the murine fossil record in order to identify reliable and suitable primary fossil calibrations for this subfamily, and to provide a reliable estimate of the timing of diversification of Murinae subfamily using multiple fossil calibrations, and also to know their biogeographic history of the family. So this study will give us a robust phylogeny, which is crucial to infer temporal time frame and so and that temporal time frame which indicate time of origin and diversification of the most major lineages of Murinae subfamily. Phylogenetic studies based on molecular data solely tells us about divergence of different lineages based on molecular clock, which does not consider the heterogeneity in rate of evolution. The rates of extinction and origin can only be reduced if calibration is done with the help of fossils. So the importance of paleontological data in studies has been described by Ryan Grant, and that states that molecular phylogeny makes only sense when it is unified with geological and paleontological data. And phylogenetic analysis will be a trustworthy estimate of timing of diversification of the family using multi-locus data sets like molecular and morphological data, and to lean on the resulting naked phylogeny to reconstruct the biographic history of the family using up-to-date analytical approaches. So to calibrate this subfamily, most of the fossils were collected from Miocene age of Indian subcontinent, and all these fossils are from Shivalik region, which is on the boundary of Pakistan and India. So these all regions are also primate-bearing sites. So the ages of these rodents are important to mark those primate-bearing localities on geological timescales. So these all the rocks are here from Neogene to Quaternary age. So now moving to the methodology part. First thing we do, we collect sediments from the field, and maceration was done of the bulk material. Fossiliferous sediments brought from the field and soaked in water and acetic acid and or in kerosene and water, depending on the hardness of the sediment. After which loose material was put inside a set of different sizes of the sieve, and macerate was passed through them. And then material collected in the sieves was dried and then observed under binocular microscope for the collection of fossils contained in the sieve. Each collected specimen properly labeled and catalogued after the collection for taxonomical studies. So for molecular data, I have selected 37 species, in which 5 species chosen for outgroup as an outgroup taxa, or and 332 were of Murinaceae family. So all the data, molecular data of 15 genes collected from GenBank, and Genius software was used for the sequence alignment, and Mesquite software used for creating access file, and all these analyses were performed on the Cypress Science Gateway, in which I have used Mr. Bayesian software, and Mr. Bayesian was performed for 400 million generations. And to know the best partition model, J model test was used, and that gave us GTR and HKY model. To view the results, PIC3 was used, and then dating of that Mr. Bayes tree was done on these two software. And for paleobiography, I have used BioGeoBears on our environment. And these are fossil calibrations which were used on the tree, Antimos chingiensis, which is 13.8 million year old in Pakistan region. And dating method was magnetostratigraphy. And the next one was Parapodium vagile, which is 10.5 million year old. Another one is Carnimata darwini, which is 9.2 million year old, and Moss 8.0, Lithomis and Herbicanthis, which is 6.1 million year old. So this figure showing dental morphology of murine rodents, based on which Murinaceae subfamily is divided from Chrysitinae subfamily. So this is the enterostyle in this figure you can see, which is not present in other subfamilies, and this is the diagnostic character of this subfamily. But due to convergent evolution, most of the species showing repetition of the morphology in younger species also. So it was difficult for us to give evolutionary tree based only on morphology tree. So that's why we tried to include fossils in molecular tree. So these are the results. This is huge data, so it might not be visible to all. So it's showing 13 subfamily, 13 tribes of subfamily Murinae, and on our this is the biogeography results, which were showing 7 biogeographic realms. Based on our analysis, what I found, that Murinae have 13.8 to 14.8 million year age, which is very close to fossil data, because we have found 13.8 million year old oldest fossil of this subfamily, and previous authors gave broad ages from 12 million year to 23, which is not for Murinae, but those ages were for Muridae. And my thesis work is focused only on Murinae subfamily, and my work is different from them because I have used uh, most of the species, around 50% species from Murinae, and other people have used only 150 species, or yeah, they have used 150 or 200 species of Muridae family. So their focus was not Murinae. So based on my results, Haplomyini is diverged from Phenomyini around 13 million year old, uh, and Phenomyini around 8.7 million year, Ratini around 8.8 million year, and all these dates are here. In biogeography, I observed that Murinae started diversifying from India to Africa around 11 million year ago. So we can say that at that time Africa and Eurasia was joined because sea was dry, and that become the path of diversification and movement of these uh, marine rodents from Eurasia to Africa. So that's all I have.
All right, so do we have any questions for Rohit?
Sorry, I forgot to.
No worries. So with you still got some time left, what do you plan for a follow-up for this? What do you want to do next?
Uh, Actually, I want to extend this work from subfamily to family level, because in this family there are five subfamilies in which morphological there is no morphological data importance in any phylogenetic tree. So I can try to code morphological data for those subfamilies, other subfamilies, and I can try this tip dating method on those subfamilies.
All right, anyone else? Well, thank you, Rohit. Our next speaker is Azil, but he was a little worried about his internet connection, so he actually sent us the recorded video for us to play here. So our volunteer is the one who has that available. We'll hit play once it hits 1:45.
Hello, good morning. I'm Esel Galindo. I present the work, the manifestation of hybrid sterility requires a greater number of sterility genes in the heterozygous introgressing segments than in homozygous introgressing segments. Background. In our previous study in press virtual presentation CS78 of Blanquestela Chavez Sandoval, Sydney 1996, for the first time introgressive segments in a homozygous state of the pair is spaces D quefere were obtained in the D gusati. Unlike heterozygosity, introgressive segment that only produced male sterility. Homozygosity, introgressive segments producing male sterility or sterility in both sexes. All these allow us to infer a genetics hybrid reproductive isolation architecture for the pair of sister species constituted by the heterogeneous distribution of the chromosome of large female sterility segment that include at less on smaller male sterility segments, where sterility is directly produced by heterozygous or homozygous recessive for a minimum their salt size. For male heterozygous integration, they require a minimum size for approximately 30%, and for mild integration that no sterility effect. While for mild homozygous integration, a minimum size of 2% is required, while for female integration, a minimum size of approximately 15 is required. The synthetic table of the chromosomal arm of Muller elements A, B, C, D, E, F show the phylogenetics relationship of the 12 sequence species of the Drosophila. On the left, on the figure and the diagrammatic karyotypes codes by colors. On the right, gray and the and red rectangle, the synthetic relationship between the chromosomal arm of the D. melanogaster, while D. mojavensis are the show. D. mojavensis is a species so close to D. busati that is shared high homology of chromosomal 4. From the result and the review, we the literature on the introgressive segment sterility among the sister species pair D. quefere and D. busati. We conclude that mild heterozygous introgressive segment sterility occurs by meaning their salt size than start in the 20% and female the is no sterility effect. While male sterility in homozygosity occurs by meaning their salt size of the 21% and the female of the 15.40. This has allowed us to infer that each segment of the female sterility in homozygosity is larger and the iguals included and segments of male sterility that is smaller. Methods. In the bioinformatic study carried out entirely by hand based on the genomics maps of the busati, D. mojavensis, and D. melanogaster, and the annotation on the genes in the platforms of the Drosophila busati consortium, the place base and the gene band. We identified and the genes present in the six homologous introgressive segment on the previous study. Each of the genes were classified according to the association with mutant allele, than sterility, lethality, and the other visible mutants phenotypes. The total numbers of the genes associated with each type of alleles of particular mutants phenotypes, sterility, lethality, and other visible phenotypes is shown for each strict chromosomal region corresponding to the six introgressive segments. Table 1 and graphic map. Each bar represents the number of the sterility genes with the respective associated mutants alleles. The blue bar represents the number of sterility genes. The orange bar represents the number of lethality genes. And the green bar represents number genes for other mutants phenotype visible. In the old case is observed that with the increase in progressive side, the number of mutant genes of the age state, sterility, lethality, and other phenotypes increase. Numbers sterility genes with the specific function in oogenesis, spermatogenesis of process for each strict chromosomal region. However, the total number of the genes functional in the testicle 16 is lower than the number of the genes that function in the ovary 99. Only 30% is dedicated to oogenesis and the spermatogenesis. 30 and 18 genes respectively. However, only 50% and the genes dedicated to the oogenesis can mutant to the stability phenotype, 15 genes, while 20% of the genes dedicated to the spermatogenesis can mutate to the sterility phenotype, 9 genes. This explains why explain the fact that in the most previous studies and the greater number of sterility genes have been indefinite. In this study, we revealed in the contradictory way that there are more than with as many fertility genes than can mutate to female sterility than kind mutant to male sterility. Conclusion. We conclude that there are most Quincy as many oogenesis genes than the can mutant to sterility, compared to the number of spermatogenesis genes than the can mutant to sterility, which are heterogeneously distributed. There are a minimal number of genes needed to produce heterozygous sterility. Males among 41 spermatogenesis genes. Female and last 271 oogenesis genes are then needed to be placed in the heterozygous recessive state to produce the respective sterilidad of each sex. There is a minimum number and genes needed to produce homogeneous sterility made between 311 spermatogenesis genes. Female at last 20 oogenesis genes are the need to be placed in the recessive homozygous state to produce the respective sterility of each sex. There are the greater number of the female sterility genes than the male sterility genes. This masks the identification of the number of the female sterility genes. In addition, it produces more robust female fertility, which explains why the great effect of sterility is suffered by segmental males. Thank you, team. Es el Jacobo Galindo Pérez, Blanquestela Chávez Sandoval, Francisco García Franco, Liliana Baradica Cañón. Thank you very much.
And we still have plenty of time for questions. So any questions we have here for Isel?
Hi, I guess I can jump in. Would you say that this trend of twice as many fertility genes in females is related to the sex determination system in Drosophila? I can type as well. That's easy.
Bueno, no se encuentra determinado por el sexo esta cantidad de genes, sino principalmente por la posición de los genes en cromosomas. Por los genes
que son
se pueden encontrar,
que son capaces de mutar a
bueno, que pueden mutar a esterilidad y son capaces de mutar a esta esterilidad.
Oh, I see. Okay, great. Thank you.
If you had the time to expand this, would you prefer to expand into other populations within the same species or other species that are phylogenetically nearby?
Sí, desde luego se podría trabajar dentro de todas las especies, principalmente del grupo de Drosophila, pues hay muchas centenias dentro de los cromosomas, entonces se podría trabajar con todas las especies y con otros géneros podría ser también.
We've still got another minute. Anyone else have a question here for our speaker? All right, I think then we're going to thank you again for presenting, and then we can set up so that our next speaker has the chance to share their screen.
Muchas gracias.
Hello, so I'm going to share my screen. So do you see well my screen?
Yes, we do.
Okay, perfect. Also,
We're just going to have you wait just a minute until normal start time because people might be moving back and forth between the different rooms.
yeah, okay, no problem.
All right, you can go ahead and start whenever you wish.
Okay, so good afternoon, everyone. I'm Christelle Panager, and I'm very glad to present my results on my Master 2 internship that I made on the impact of seasonality on predation pressure and butterfly communities at different elevations. So first of all, I wanted to give some context. In fact, it's in the neotropics that we find the highest diversity on Earth. We have an extensive literature on the subject, and many hypotheses have been proposed to explain this phenomenon. For example, it might be explained by the climate stability or the partitioning of ecological niches. However, we have only few studies on the impact of seasonal changes, yet there could be effects on species communities or interaction with predators. So it would be very interesting to study these seasonal changes in the neotropics, for example, on the Ethomini community and the Ethomini tribe. This tribe comprises many toxic butterflies that are also aposematic, common and diverse in the neotropical mimicry rings. To be short, aposematism corresponds to the presence of signals on individuals to warn a predator of its toxicity. So, for example, in the Ethomini tribe, we have the presence of colorful patterns on the wings that inform a predator of the toxicity of the butterflies. So here I wanted to show what are the mimicry rings. Here you have an example of mimicry ring in which we have Malayan mimics. Here you have two different species that are both toxic. However, they look very alike. And then you have the Batesian mimics that also look alike the Malayan mimics. However, they are completely edible. And so here I represented examples of different mimicry rings. So here you have 4 different mimicry rings. I based my internship on a study that was made previously in our team. And we discovered that the Ethomini tribe was more abundant and diverse with altitude, and it was correlated with lower temperature and rainfall. However, we didn't find any difference in predation pressure with altitude during the dry season. So I wanted to see if we had similar patterns during the wet season, and also if there was seasonal variations in Ethomini communities. So in this perspective, my first question was, how do seasonal variations affect Ethomini communities at different altitudes? So I expected that we would have a greater abundance and diversity during the wet season, because it's during this season that we would have more resources for larvae and adults. And so it will help Ethomini communities to increase in abundance. And this increase will allow us to detect them because, for example, rare species would be more abundant. And in this case, we would detect a higher diversity in the environment. Also, we would wonder, how do the seasons affect predation pressure at different altitudes? And so we would expect more predation pressure during the wet season, because it's mostly during this season that we have many species that mate and nest. So in this case, we would expect more predation on butterflies to feed the young. To sample the communities, I've been to 3 different sites in French Guiana during the wet season, and I compared what I sampled with that of the dry season we sampled 2 years ago. And so I've been to 3 sites of 3 different altitudes that are the Somaripa for lowest altitude. Then we have the Caux Mountain for the medium altitude, and finally the Galbao Mount for the highest altitude. At each site, I sampled as many butterflies as I could. We were 3 people to do it, and we collected Ethomini, but also Malayan mimics and Batesian mimics. And based on what we got, I calculated the diversity indices and tested if the communities were different as a function of the seasons and sites. Then, regarding the predation pressure, I made 200 artificial butterflies per site. They were all based on the reference of Pierrella lamia. You have an example here. And Pierrella lamia is a very common cryptic and edible butterfly in French Guiana. We put them at 2 different heights, one close to the ground and one at 2 meters high, because we wanted to test if the predation rates were different between the model heights. Basically, it's because in the literature, we can see that the butterflies are vertically stratified in the neotropics. So it would be interesting to see if this vertical stratification will be potentially due to differential predation rates. And so I focused on the predation from birds because they are the main selection pressure on the aposematic patterns in butterflies. So to speak about my first results, here I wanted to show the rarefaction curves of the Ethomini communities as a function of altitude, but also seasons. And what we could see is that with an increasing altitude, we had more species, so the y-axis, and more individual per species, it's the x-axis. So with an increasing altitude, we have more abundant and diverse communities. Also, for 2 sites out of 3, we don't have very much differences between the seasons, while for our lowest site at Somaripa, we have a great difference between the seasons. That is, during the dry season in yellow, we have a low number of species and individuals, whereas during the wet season in blue, these numbers increase. For the predation pressure, I wanted first to measure the flight heights of different butterflies and see if they would vary as a function of mimicry rings or a category of butterflies, for example, Ethomini, Merlion mimics, and Batesian mimics. And what I could find is that mimicry rings, here on the x-axis, have different flight heights. Also, within one mimicry ring, for example, Mamercus, we have differences between the different butterflies. That is, Batesian mimics in green lie significantly lower than Ethomini in orange and Merlion mimics in yellow. However, Ethomini and Merlion mimics fly about at the same height. Even though we had significant differences in flight heights, I didn't find any difference in predation pressure with model heights, seasons, sites. Also, I didn't find any significant interaction between the seasons and sites. Also, we found very low predation rates by birds in French Guiana compared to other literatures. To discuss my results, I found a higher diversity and abundance of Ethomini at high altitude. This might be potentially correlated with lower temperature and rainfall, like we found previously in our team. And also maybe at high altitude, we could have more stable climate, for example, in the cloud forest, in which the temperature and rainfalls are more constant throughout the year. So in this case, it would create like a refuge zone for many species, for example, rare species, and thus it would allow them to thrive in these conditions. However, we found only at the lowest altitude a seasonal variations of the communities, and more precisely, a mild increase in diversity and abundance during the rainy season. So it may be because at the lowest site, we have a more drastic difference between the seasons climatically. And also maybe during the wet season, we would have more favorable conditions like lower temperatures, allowing the population to grow. However, I didn't find any evidence of seasonal and altitudinal variation in predation, so it might not be the cause for spatial and temporal variations in ethnominy abundance and diversity. Regarding the flight heights, I found that butterflies from different mimicry rings fly at different heights. So here, as an example, I showed two different mimicry rings, one in orange and yellow, and one that is quite transparent. And so I also found that Batesian mimics here that are completely edible fly significantly lower than the other butterflies, that is the Thomini and also the Malayan mimics. Here they are all toxic. However, I didn't find any vertical variation in predation pressure, so maybe it's not the cause for vertical stratification in Ethnomini flight heights. So in a perspective to this study, I'm currently investigating if we can score the wing damages to better estimate the predation pressure by birds on aposematic butterflies. And it will allow us to see if the predation of specialist predators varies spatially and also temporally. Also, it would be interesting to see how environmental factors vary with seasons as a function of altitude. And so it will be useful to confirm if the climate is indeed more stable at high altitude, which would explain why we have more stable communities between the seasons for these altitudes. Thank you for listening. I thank also all the people that helped me during this internship, and I will be very glad to answer the questions. Thank you.
Thank you. And we, of course, have plenty of time for questions. So raise your hand. We are happy to call on you and have you call out a question. Mhm. All right, I will ask one then. So you did this during your internship. Obviously, you don't always have time to get everything done you'd want to do. What else would you like to see done with this system?
In fact, actually, I only had the time to see the differences for 3 sites, but in single time, it's 1 site per altitude. So it would be very interesting to duplicate the sites. In the previous study, they went to 4 different sites for each altitude. So it would be useful to do the same, to have enough replicates for all the altitudes.
All right, if we have no more questions, we're going to thank our current speaker. And now we actually have a little bit of a